Hi everyone, I'm excited to talk to you today about my work on low coverage sequencing and how it cost effectively detects known and novel variation in underrepresented populations. As a geneticist, I like to start at the beginning. For humans, that means we need to start in Africa, where humans originated on the order of hundreds of thousands of years ago. As humans migrated out of Africa into the Middle East, into Eurasia, and subsequently into the Americas, at each of these migration points, they took a subset of genetic variation with them. That means that the average out of Africa population has about 1 million fewer variants than the average African population. This human history over the course of the past 100,000 years or so ago informs the landscape of genetic variation across the world and is important to consider when we design genetic studies. Speaking of genetic studies, we also know that they have vast Eurocentric genetic study biases and that this is a huge problem. Right now, 80% of participants in GWAS are individuals of European descent, which is far out of step with the global population, where they comprise about 16% of the world's population. Perhaps even more troublingly, if we look at the trends over time on the bottom part of this chart, you can see that the fraction of individuals with primarily non-European ancestry peaked around 2014 and since then has stalled or slid in the wrong direction for diversifying progress and translation of genetic technologies in an equitable fashion. So on that topic, as a field, I think there are many prerequisites that we need to meet before we can execute on the most exciting, equitable, and impactful science. For today, though, I'm just going to focus on developing data and technology that maximizes data accuracy and discovery in diverse populations through new data and community resources. So I've been very lucky to be involved in the NeuroGap study spearheaded by Karestan Koenen. And in this study, we're planning on recruiting 35,000 individuals spanning Ethiopia, Kenya, Uganda, and South Africa for studies of psychosis. We've evaluated several data generation strategies across all sites. So we've genotyped individuals, including 960 individuals on the so-called global screening array. We've run 1x sequencing, and we've conducted high coverage sequencing. With GWAS arrays, most of the variants on this GSA array are most common in European ancestry populations and less common elsewhere. So the GWAS backbone here, array backbone, is actually sort of problematic because 32% of variants are monomorphic or invariant in the NeuroGap study, and even more are at low frequency. So there are big problems here. Two of them are that imputation panels are less adequate in African populations than in Europeans, and there's just more variation overall in African populations because humans originated there, and this variation is more often untagged. Low coverage sequencing has been proposed as a strategy that might capture genetic variation in a more unbiased manner, overcoming the ascertainment issues of GWAS arrays. Companies like GenCove have worked on low coverage sequencing strategies and have shown that there are big advantages compared to GWAS arrays, especially in underrepresented populations. High coverage sequencing, which we've done in 93 individuals, would be ideal for this study, but is currently prohibitively expensive for the scale of the full scope of this project. But sequencing a small number of individuals has allowed to test different data generation strategies in pilot studies. So we've used this high coverage whole genome sequencing data to simulate for the exact same individuals what would happen if we did low coverage sequencing at various steps versus if we ran different genotyping arrays. So we use this high coverage sequencing data to either do downsampling of reads or to sample variants from the high coverage sequencing data that correspond to different genotyping arrays. So in doing the downsampling of reads, we've generated simulated data on the scale of half X, 1, 2, 4, 6, 10, and 20X depths. And for the different GWAS arrays, we've looked at the global screening array and several different arrays with varying densities. And so with these different strategies for low coverage sequencing, we need to do what's called genotype refinement. So here we can use the genotype likelihoods rather than the hard genotype calls themselves, and reference haplotypes like those that we can derive from the 1000 Genomes Project to better estimate the genotype state. And then for both low coverage sequencing and for GWAS arrays, we can use imputation to sort of fill in the gaps, which improves the number of variants that we have called in our data set. So one really big advantage with low coverage sequencing data is that we can actually detect rare variants. So we're sensitive to detecting these variants with whole genome sequencing data where we actually can't identify these in GWAS arrays. And this is really important. Colleagues like TJ Singh and others in the PGC have shown that there's really big important differences in terms of what we're finding and the scale of the effects in rare variant studies versus in common variants. And we need something to bridge this gap to enable a larger scale of genetic studies to be conducted so that we're finding the entire variants across the entire genetic architecture of these diseases. So low coverage sequencing actually facilitates and bridges that common versus rare variant architecture gap. At 4X sequencing, for example, we're detecting almost 50% of the singletons that are present in the full coverage depth data. And at 4X depth, we're also sensitive to detect 95% of the variants present in the full depth data set. Notably, GWAS arrays can't capture those singletons unless these are on the array backbone themselves. And they can only capture those genetic variants that are either imputable or on the arrays themselves. So they only capture 62% of the variants that are common in NeuroGap because NeuroGap is not well represented in the 1,000 genomes reference data. So this is the summary of the results of data quality that we found. So in the left panel, we're seeing the raw data that came out of GATK or a data science platform. In the middle, we're seeing the refined genotype variants from the low coverage sequencing data. And on the right, we're seeing the imputed variant calls. So on the right, we have a comparison of both the different GWAS arrays that we compared, as well as the low coverage sequencing data. And so what you can see is that at about 1x sequencing, we're doing similarly to how we're doing with the lower density arrays like GSA and the Psychiatric. And at 4x sequencing, we're outperforming all of the commercially available array data. So this means that we're doing a really good job with 4x sequencing. Now, what about price? So we see that 1x and half x sequencing are less expensive than these low density arrays like the GSA array and Psychiatric, and that 4x is competitive with other high density arrays. I would like to point out that there are a few caveats with this price list. So they don't include things like bulk discounts. These are list prices that are publicly available from Illumina. And with a large sequencing center, you might get discounts because of the high volume and throughput of the sequencing that these centers are doing. These costs also don't include library construction or labor costs, although these should be pretty similar between sequencing versus genotyping. As key takeaways here, low-pass sequencing at 4x depth or greater especially outperforms GWAS arrays pretty seriously in underrepresented populations in the following ways. It identifies novel variation at all depths, including rare variation that GWAS arrays are insensitive to. It captures other forms of inaccessible variation, including structural variants, or for saliva samples, microbiome information, and other forms of variation that we might be interested in. It's also cost effective, especially compared to high density arrays. And it facilitates the creation of critical, missing, and persistent genomics resources like imputation reference panels or haplotype resources. So I'll stop there and say thank you to all of my collaborators, especially those at Broad or ATGU, those involved in NeuroGap, those in Ginger, which I didn't have a chance to mention, but that's part of this as well, the genomics platform and my funding.
Hello, my name is Bizu Gelaye. I'm an assistant professor of psychiatric epidemiology at Harvard University and Mass General Hospital. I'm also a member of the PGC PTSD working group. I'll be sharing with you recent work that we completed with my colleagues looking at polygenic prediction in GWAS of 3 psychiatric phenotypes in Peru. This was a multidisciplinary work and collaborative work done with my colleagues Hanyang Shen, Hailiang Huang, Marta Rondon, Sixto Sanchez, and Laramie Duncan. As many of you know, human genetic studies have historically focused on European ancestry participants. Even though there are some encouraging efforts underway, we still have a long way to go to address the legacy of Eurocentric bias in genetic studies. As has been the case with many genetic studies more broadly, there have been few GWAS studies and PRS studies of psychiatric outcomes in South America. Even though Latino and Hispanic samples from the US are most similar to admixed populations in South America in terms of their ancestry, there is great heterogeneity when it comes to exposure to risk factors and living conditions. And psychiatric phenotypes are deeply rooted in cross-cultural context. Therefore, we conducted the present study, which is the largest psychiatric genetics studies to date in South America, to look at polygenic risk scores of depression, PTSD, and suicidal ideation. We also conducted a GWAS to make summary statistics available to researchers worldwide. This was a result of multidisciplinary effort and collaborative work with partnership with the Peruvian Ministry of Health, as well as one of the largest hospitals, La Maternidad, or Instituto Nacional Materno Perinatal, where we recruited more than 3,000 women who were attending prenatal care clinics prior to 16 weeks of gestation. We used structured interviews to ascertain the psychiatric phenotypes, and blood samples were used for genotyping. We assessed PTSD using PCL civilian version, using a Spanish language and validated for this population. We used the Patient Health Questionnaire 9, the Spanish version, to look at depression, and the item 9 to assess suicidal ideation. Initially, the work was supported and funded by NICHD. The genetics work was supported by Koenen Veterans Bioscience and Stanley Center for Psychiatric Genetics at the Broad Institute. We used the Illumina multi-ethnic global chip for genotyping, and we obtained summary statistics from the PGC working groups from PTSD and depression working groups. These are the results showing the ancestry clustering based on our study, the PROMISE study, in the 1,000 genomes. On the top panel, we can see the 3 principal components of the PROMISE sample and the 1,000 genome sample plotted together based on principal components computed upon the combined sample. As one could expect, the PROMISE samples, which are depicted in magenta color, clustered with the pale 1,000 genome samples, which are indicated in golden triangles. Not surprisingly, most of the PROMISE participants aligned well with the 1,000 genome pale sample, who are Peruvians from Lima, Peru. A small number of PROMISE participants dispersed among other samples from populations in South Americas, and this was also the case for a minority of the pale samples. The bottom panel B shows the top 3 principal components from the PROMISE participants alone with PTSD cases and controls. Here are the results of the polygenic risk score prediction for the 3 phenotypes using 2 things. The top panel uses depression based on polygenic risk scores, depression-based polygenic risk score, and the bottom panel shows PTSD-based polygenic prediction. As I mentioned, we use summary statistics from the PGC, depression, and PTSD working groups. The main finding here is, as one can see from the top panel on the left side, depression PR race constructed using SNPs with discovery GWAS yielded the best prediction results for depression score, and they explained about 0.62 of phenotypic variance. Similarly, as shown on the bottom panel, PR races for PTSD constructed using SNPs with discovery GWAS significantly predicted PTSD and explained less than 1% of phenotypic variance. As we all know, there is issue of confounding by population stratification, so we conducted a sensitivity analysis to account for population stratification, and the results remain the same. Finally, here are the Manhattan plots for the GWAS results. As one could expect from an underpowered study, the results were null with respect to identifying individual loci. But we're making the GWAS summary results available to increasing access to non-European ancestries so that we can facilitate future meta-analysis. In conclusion, to our knowledge, this is the first report to provide psychiatric polygenic scoring and GWAS results from a large sample from South America. Our results show that polygenic risk scores derived primarily from European ancestry and non-admixed populations could potentially apply and validly predict psychiatric phenotypes even from individuals who are considerably different in terms of their environment, culture, and ancestries. This means that genetic influences of depression, PTSD, and suicidal ideation are at least partially shared across populations. And this work is consistent with other studies that show polygenic results from the literature looking at depression and PTSD polygenic risk scores are predictive of psychiatric outcomes in populations of other ancestries. By sharing GWAS results from the PROMIS sample, we feel like this is an important contribution and can be used in other populations. As has been the case in the past, more PRS studies are needed to replicate our study findings, as well as to include other populations from diverse ancestries across the globe. We're indebted to participants of the PROMIS study, as well as the funding agencies that made this work possible. Thank you.
Hi, my name is Rocky Stroud, and I'm going to be presenting today on our research study, neurogap psychosis. So neurogap psychosis stands for the neuropsychiatric genetics of African populations. And this was really launched to address the global disparities in neuropsychiatric genetic research, particularly in Africa. As many of us know, the field of genetics is not very diverse. And we are, in this study, aims to expand the diversity of global collections. So this study is run by the PIs shown here, Dr. Karestan Koenen out of the Broad Institute and Harvard School of Public Health. And our research is in East and South Africa. So we have Dr. Solomon Tefera out of Ethiopia, Dr. Dickens Ikenna out of Makerere University in Kampala, Uganda, two Kenyan sites, one with Dr. Simon Kariuki out of Cameroon Wellcome Trust, which is on the coast of Kenya, and Dean Lukoye Atwoli at Moi University in Eldoret. And then in South Africa, we have Professor Dan Stein out of the University of Cape Town and Dr. Zuki Zingela out of Walter Sisulu University, which is in the Eastern Cape. So we are collecting in both Eastern and South Africa, but we not only are expanding diversity by adding these 4 countries, but we also are expanding diversity within countries, as we know that there's a lot of diversity within countries as well. So you can see in Uganda, we have 4 different collection sites from both North and South, and then also in South Africa, as an example, we're doing both Eastern and Western Cape, as the diversity within countries is also quite different. So our sample collection projections and what we aim to do by the end of 2022 is to collect 35,000 samples. So the breakdown here shows that our study is a case control. So anyone deemed to have psychosis as the umbrella term. So that could be schizophrenia, bipolar disorder, schizoaffective, mania, etc. And then also matching controls. So in Ethiopia, we're aiming to do 8,000, Kenya 9,000, South Africa roughly 10, and then Uganda 8,000 as well. So we have a pretty in-depth process. We collect about an hour and a half's worth of phenotyping questions, but we also collect a sample. A big part of our study is also to ensure the sustainability for future research within countries in Africa. So the sample is collected and we do the DNA extraction in country, and then 1 aliquot is sent to the Broad to do for us to eventually do the GWAS study that we will be conducting, and then the rest of the sample is actually left at those universities or research institutes for the creation of biorepositories in country. So to date, as of October 1st, we are at 19,000 of our projected 35,000 goal. And this chart shows both participants recruited from our original start, which was back in June of 2018, to now as well, and that's in blue. And then in orange is the samples that we've received here at the Broad Institute. And we expect as I said before, to reach our target hopefully by 2022. So by the numbers, it took us 2.5 years to get our protocol approved. And that is because we actually work with 12 different IRB committees. We have both here in the US, but also in our collaborating and partnership institutions in Africa, have to go through a very rigorous process of getting all of our documents and things approved. And we have a very large staff. So there's 42 research assistants and project managers associated with the study, working day to day to collect and recruit and enroll our participants. So where are we heading? So we're going to continue recruitment, and we have had to update all of our protocols to safeguard both research participants and our staff against COVID-19. We took a lot of steps to procure PPE, as well as update our protocols to ensure that we were to ensure that we were protecting our participants and our research staff. We're going to be doing a validation study on the psychometric tools and local languages. Our study right now is being conducted in 8 different languages, and we're going to be validating our tools. We use the MINI, which is the MINI instrument, International Neuropsychiatric Instrument, and we're going to validate that with the SCID. We recently were awarded a part of the U01 grant at the Bird Institute at Harvard, as well as other collaborating institutions. And a big part of that will be developing the genetic analysis for past sequencing, which Alicia Martin will be speaking about, as well as expanding our collections in Ethiopia. And then we, as always, are trying to ensure that we are doing capacity building at all levels. So not only do we have the Ginger program, which you'll be hearing from Emmanuel and Mwesiga, but we also do capacity building at the lab, at the program manager level, and at the research assistant level. And we really want to ensure that we leave behind a sustainable research endeavor. So I wanted to acknowledge the program managers. So these are the 5 program managers that we have working in countries, and without them, and they really are the backbone of the study. Without them, we would not be as successful as we are. And then these are obviously acknowledgements for all the people on the team. Many people are missing, but really wanted to say thank you to all of them as well, and say thank you so much. I hope everyone can stay safe. Thank you.
Hi, my name is Elizabeth Atkinson, and I'm an instructor in the Analytic and Translational Genetics Unit at Massachusetts General Hospital. I'm excited to tell you today about a method I've been developing that lets you include admixed individuals in genome-wide association studies in a well-calibrated manner. To start off, the vast majority, almost 80% of GWAS, are conducted on European descent cohorts. Only a few % of all GWAS include individuals of admixed ancestry, despite these groups making up more than 1/3 of the US populace. This issue is what prompted us to want to develop a method that could allow us to include such populations in a well-calibrated genome-wide association study, either alone or alongside homogeneous cohorts. So the way that admixed groups are currently handled is generally by using principal components analysis. So on the slide here, I'm showing the PC plot for the Psychiatric Genomics Consortium PTSD working group. And often the first step in large-scale collection efforts is to define who is homogeneous enough to include in a GWAS. So we'll generally draw a circle around people homogeneous enough to include and exclude everybody else. And this, in general, will include the European descent individuals, as they most often represent kind of the bulk of the samples. Admixed individuals specifically are discarded usually due to concerns over population structure, as if there is population structure, this can bias your results by resulting in false positive associations. So the way that we handle this issue is by incorporating local ancestry. So local ancestry tells us the ancestral origin of each particular haplotype tract in an admixed individual at each spot in the genome. So in this example, three-way admixed individual, the y-axis represents the autosomes. The position along them is shown on the x-axis, and because we're a diploid, the top and bottom half of each chromosome are painted differently. So each tract in this individual is color-coded according to the ancestry from which it derived, and each person in the cohort will have a unique fine-scale pattern of local ancestry. So the intuition behind Tractor is that to correct for population structure in admixed people, we effectively scoop out the tracts for each component ancestry. So we literally do this by outputting new VCF files with other ancestries blanked out as missing data, but also in the sense that we output ancestry dosage files, which will track the ancestry context of each of the alleles in each person at each spot in their genome. I've also built a couple additional features into Tractor that I won't have time to touch upon here, such as using local ancestry to correct for long-range phase errors in admixed cohorts. So the statistical model that we've built into Tractor tests each SNP for an association with the phenotype by splitting up the ancestry context of the minor alleles. So in the two-way admixed example, we include terms for how many copies of the index ancestry there are for this person at this spot in the genome, as well as how many minor alleles are falling on the first ancestry backbone and the second ancestry backbone. So this is what corrects for the fine-scale population structure. If there are differing allele frequencies for the ancestries at that position, they won't be confounded as we'll have deconvolved them. This model can also scale to an arbitrary number of ancestries and allows for the ready inclusion of all of your necessary covariates. To evaluate Tractor's power to discover GWAS loci, we ran sets of simulations varying the parameters of the effect size difference across ancestries, overall effect size, minor allele frequency, overall, and difference across ancestries, admixture fractions, and disease prevalence. I'm just showing 4 of the major models used in the paper here. In all of the different panels, the dashed line is our Tractor model, and the solid line is our standard GWAS model, both involving 10 principal components. You'll notice that the Tractor model often has boosted GWAS power, meaning we would have the power to find loci that would have been missed in this admixed cohort using standard GWAS procedures. This power boost is the most dramatic when there's a large effect size difference across ancestries, as shown in panel D. This is the case when there's an effect only in one ancestry on the European tracts in this two-way admixed African-European cohort. Notably, when referring to effect size, I mean both to the case of when there's a difference in the true causal effect across ancestries, as well as when there's a marginal effect difference across ancestries. So this perceived marginal effect size of a tagged SNP might be different across the groups depending on the minor allele frequencies and the LD pattern that's resulted from that population's demographic history. So this means we should expect to get gains in GWAS power at a large number of variable positions across the genome. Additionally, in cases when we would not expect any value from incorporating local ancestry, that is, when everything is identical across ancestries, we do take a small hit in power from the inclusion of an additional term in our model, but this hit is not too substantial. Finally, the last thing we wanted to confirm in our simulations was that Tractor not only added power to GWAS to discover real loci, but also was able to produce accurate ancestry-specific effect sizes. And indeed, for every genetic model we tested, it produced highly accurate ancestry-specific effect sizes. So to close up, I'll remind you of the primary deliverables of Tractor. First, I've developed a readily implementable analytic framework that allows for the well-calibrated inclusion of admixed people in large-scale collections. So you no longer need to toss out all your admixed folks. You can include them either in a well-calibrated GWAS in your admixed cohort alone, or in a large-scale study with your homogeneous collections. We've also added an optional step to recover long-range tracts that are disrupted by phasing errors. With respect to GWAS, our novel local ancestry-aware GWAS model can improve your results in a variety of ways, most notably through boosting GWAS power, also through generating ancestry-specific effect size estimates, as well as helping to localize GWAS signal to be more close to the causal variance. So in sum, this framework should dramatically improve the existing methodologies for studying admixed individuals and allow for a significantly better calibrated understanding of the genetics of complex psychiatric disorders, in underrepresented populations. So with that, I would like to thank you very much for your interest. And if you'd like further details about our method, including assessing its performance on empirical data, please read our preprint, which is currently available on bioRxiv. Thanks very much.
Good day. My name is Lerato Majara, and today I'm going to be sharing with you results from a project that I've been working on that shows that polygenic risk scores have low generalizability in African populations due to genetic and environmental factors. About 80% of genome-wide association studies, or GWAS, are conducted in populations of European ancestry, compared to the 2 to 3% in African ancestry samples. And polygenic risk scores, or PRS, are computed from GWAS summary statistics. Now the problem is PRS prediction accuracy decays with increasing genetic distance from the discovery cohort. And what that means is if polygenic risk scores are computed from European ancestry samples, prediction accuracy is the highest in European samples and lessens by 4.5 times in African populations. So the aim of this study was to investigate how PRS generalize within and among diverse African populations. And the reason we're wanting to do this is because Africa is the most genetically diverse continent. And in fact, the genetic divergence across Africa is comparable to that across out of Africa continents. So to achieve our aim, we used 2 sets of data. And the first was individual level genotype data from the general population cohort, GPC, comprising 4,778 individuals from 9 ethno-linguistic groups in Uganda. We also used the UK Biobank, comprising 350,000 individuals from the United Kingdom. So within the UK Biobank, we used the European ancestry samples as discovery, and we used the diverse samples as target to assess the transferability of polygenic risk scores. The second set of data was GWAS summary statistics from GPC, Biobank, Biobank Japan, and PAGE, which largely comprises African American and Hispanic and Latino participants. We assessed 34 quantitative traits across these cohorts, spanning anthropometric, blood factor, blood pressure, glycemic control, and liver function measures. So after computing polygenic risk scores in 10,000 European ancestry samples from UK Biobank, we observed variable prediction accuracy across continental ancestries, with Africa having the lowest prediction accuracy. What was even more interesting was that when the African group was broken down by region, there was variable prediction within the African ancestry, across African ancestries, with prediction accuracy being the highest in the admixed group and lowest in the Southern African group. We also observed a 2-time difference between prediction accuracy in Ethiopia compared to the Southern African group. When we looked at prediction accuracy in the Uganda cohort, there was low prediction accuracy in general, but there was also a difference in prediction accuracy in the Uganda cohort compared to the Eastern samples in the UK Biobank, consistent with cross-cohort difference in prediction accuracy. So then we wanted to assess the impact on prediction accuracy when the discovery and target cohorts were matched for ancestry. And we did this across single cohort using UK Biobank, but also in meta-analysis in BBJ, PAGE, and UK Biobank. And in general, there was improved prediction accuracy, and particularly in the America, East Asian, and African cohorts, which is consistent with the ancestries represented in BBJ and PAGE. And looking within the African group, there was a significant gain in prediction accuracy in 2 phenotypes. The first being mean corpuscular hemoglobin concentration, and the second being white blood cell concentration, WBC. And to investigate these differences in prediction accuracy, we looked at the differences in the test association statistic between UK Biobank alone and the meta-analysis cohort. And it turned out that there were population-enriched variants that were driving these observed differences in prediction accuracy. So for example, with MCHC, variants within HBS1L have been associated with sickle cell and beta thalassemia. With WBC, variants within the Duffy blood group have been associated with malaria resistance. So throughout this presentation, I have demonstrated 4 points. The first being that African genetic risk is inaccurately inaccurately predicted using European GWAS summary stats. The second was that cross-ancestry prediction diminishes accuracy more than cross-cohort. Third, that inclusion of diverse samples in meta-analysis improves prediction accuracy. And lastly, that large effect population enriched variants drive heterogeneity in PRS prediction accuracy. I conclude that PRS prediction varies as a function of genetic architecture, environment, and many other complex factors. These are my acknowledgements, and I thank you for your attention.
Hello, everyone. Dr. Emmanuel Mwesiga. I'm a psychiatrist in the Department of Psychiatry at Majara University in Uganda, and I am also a doctoral fellow at the University of Cape Town, funded by the NeuroGap Psychosis Study. I'm here to, in the next few minutes, make a case for why building equitable neuropsychiatric genetics collaborations is worthwhile, and I'll use myself and a case study of the Ginger program where I was a fellow for the last 3 years. So about 4 years ago, Karestan and Koenen got together with a group of other PIs across Africa, and they decided to carry out the NeuroGap psychosis study. Simply, this was a large-scale genetic study to try and understand the genetic underpinnings of psychotic disorders in African countries. We know that many of these collaborations have been made before, and we also know that many of them look this way, that people fly in, harvest the data, have a good meal, go on safari, then go home and write papers never to be heard of again. We know that this should not be perpetuated, and it should be condemned, where local collaborators have no input and have no benefit. Luckily, from the foresight of the NeuroGap study, they decided to work together with Dr. Laurie Chibnik, who is right there at the bottom of the screen. And they formed what is the Global Initiative for Neuropsychiatric Genetics Education and Research. The aim of Ginger was expanding capacity. They did not want to have safari research done, and they decided that they needed to build collaborations, train next generation of researchers, and in the long term, this will improve research capacity and reduce the burden of serious mental illness through research. They took a chance, and I agree it was a big chance, on 17 fellows, diverse backgrounds, different countries, different levels of training, and just a group of very interesting people. So we had 6 psychiatrists, 7 geneticists, but in there we also had a clinical psychologist, we had a pharmacist, we had 2 epidemiologists, just to show the diversity of the people who were in the program. Earlier I said that they did take a chance, and I'll give an example of myself. So I'm from Uganda, and I'm a psychiatrist. And interestingly, before 2017, whenever I would go for a class and they would say SNP, I thought it was spelled SNIP, and I had no idea what it is. When Laurie would talk about R, I thought it was a letter of the alphabet, and yet she was talking about a programming language. When they would talk about working in the pipeline, I thought we were talking about oil and gas. So I was as green as they come. Maybe some of my other colleagues in this group had more experience, but the majority of us, it was just taking a chance on us. So for the next 3 years, they held our hands and they taught us neuropsychiatric genetics. You would be surprised, we started from the basics of what a gene is, what a Manhattan plot is, till 3 years later where we were, of course, again, at different levels, but able to write code, do statistics, work in a pipeline, extract DNA, and many other things. And this is what we call a true collaboration. There is now a new group of fellows, and I'm sure that they're going to take the same path and get the same benefit that we did. What NeuroGap and Ginger and Karestan and Laurie did in African terms is called Ubuntu. I am because you are. They realized that if NeuroGap or Ginger was to be great, it needed to have us, the local fellows, to also be great. So holding our hands and taking us to the next level was true African spirit of Ubuntu. Because if you look at it, in the last 3 years, so the first Ginger Fellows, the 2017 to 2020, in the last 3 years, we have had 3 PhDs started, 3 PhDs, and at the end of this year, I will have my PhD, so it will be 4 PhDs and 5 promotions, 5 fellowships. We have co-authored papers, we have done projects together. There was part of the Ginger program involved us working together with, should I say, like teachers from Harvard School of Public Health, from the Broad Institute, and doing projects of our own. And one of those group collaborations, we even had a publication in the Lancet Global Health. So the outputs were and are obvious to see. One could then ask, what is in it for the collaborators? Why would people come all the way and train these naive people in neuropsychiatric genetics research? Because they could have decided to just carry out safari research. So I put that question to one of the collaborators. Alicia Martin presented earlier, and I asked her, so what do you get coming to Dr. Mwesiga and teaching neuropsychiatrics? And this is what she had to say. She said some of the sites showed her the differences between health treatment in the US and low and middle income countries, and also her own career trajectory developed. In summary, she developed as we developed. And I think that's the beauty of the Ginger program and NeuroGap. The most important people, however, in this room and this talk are the funders. It's very important that funders are encouraged to have equitable collaborations like the Ginger program. We acknowledge that the Ginger is not the first program. We know H3 Africa has done a great job. We know Wellcome Trust has done a good job. Delta program, MEPI. But the truth is that just not enough people are taking a chance on African researchers. And this is a call. This is really the call is build equitable collaborations has a language of the NeuroGap study. 4 years ago, yes, they had to come and teach everyone how to extract DNA, how to work in a pipeline. They had to teach us statistics. But if by some lucky chance NeuroGap developed a drug, when they come back to Africa, they will be confident that they can pick Dr. Mwesiga and ask him to run a trial for the drug because they did teach me the basics of randomized trials. They can ask Alan Kalunju, who is one of the fellows, to, I don't know, extract DNA for another study. They have built capacity, they have built confidence, there's an amazing ripple effect. So I would just encourage all funders out there that building equitable collaborations is worthwhile. And if in doubt, remember 3 years ago, R was a letter in the alphabet. In conclusion, I have to acknowledge the people who funded the NeuroGap and the Ginger program. We're grateful to the Broad Institute, to the Harvard T.H. Chan School of Public Health, Addis Ababa University, where some of the fellows came from, Kenan Trust, that also had fellows, especially Dr. who is my PhD, the person who introduced me to NeuroGap and Ginger. I'm grateful. Professor Lukoya Twoli, Dan Stein, who is also my other supervisor, and Zuki Zingela. Thank you very much. And remember that equitable collaborations are worthwhile. Thank you.